Welcome back to Science 30, Unit A, Chapter 2 on genetics. And this is the second lesson in this unit or chapter, and it's on inheritance. So we're going to dive deeper into how these alleles work together to pass on these different characteristics. So these are our outcomes. Make sure you review those so you can check out what we're doing today. The new thing that you're really going to be touching on are the Punnett squares. And we're going to be using Punnett squares to figure out how the inheritance of single traits, so we're just looking at one trait at a time, and we use Punnett squares to figure out how they segregate and are passed on. We're going to cut this lesson short, and it's going to be split into two, and the sex-linked inheritance we're going to do in a second video. So there's your textbook pages. Make sure you check those out too. Okay, so let's move my video over here. Maybe this is a better spot for it. Okay, so have you ever wondered why you look more like your mom than your dad or vice versa? Well, it's all about your genes. And one quick little example is if you have a hitchhiker thumb here. This hitchhiker thumb, the ability to do that, I don't have that ability, right? My thumb doesn't bend backwards, so I do not have one. Do you? Check it out. If you do, that means you have what we call the homozygous recessive genotype. By the end of this lesson, you should know what that means. If you don't right now, that's okay. If you don't have a hitchhiker's thumb, that means you have the dominant allele. So I must have the dominant allele. So a couple terms just to review. These are from last lesson. Genetics is the study of inheritance. Heredity is controlled by genes. Genes are found on your chromosome. Each person has two genes or two alleles for each trait, one from each parent, right? And then the way that these two alleles interact will decide whether you have a hitchhiker's thumb or a non-hitchhiker's thumb. So inherited traits versus acquired traits, just for this terminology, inherited traits, these are passed on from one generation to the next generation. You can't do anything about these. You're going to get it or you're not going to get it. So some of these ones that are in humans that are very obvious to us that we can see are attached earlobes versus these free earlobes that kind of fall down. Widow's peak versus a non-widow's peak, the forehead of your ear, a cleft chin, or a non-cleft chin. Those are just some of those inherited traits. We're going to look at quite a few more as we go through. Acquired traits, so they're something that you acquire during your lifetime. So this might be a scar, or if you dye your hair, or the ability to speak 5 languages. Those you acquire during your lifetime and are not passed on genetically. All right, alleles. We're going to look at what these terms dominant versus recessive mean. And alleles are alternate forms of a gene. Okay, so genes will come in two forms for the genes that we're studying. So you're going to inherit one allele from each parent. And the way that these two alleles interact is going to determine how that trait is going to be expressed. So for example, it's a lot easier to look at an example when you're talking about this kind of thing. The gene for flower color. The alleles can be found for purple or white. So in this case, you'll see there is the allele for purple on one chromosome, and there is the allele for white flowers. So this individual plant in their cells has both these chromosomes, one from one parent, one from the other, and how these two interact are going to depend on the dominance of these alleles. So the dominant allele is masking the effect of any recessive allele that's present. So when we talk about a dominant allele, we use a capital letter to kind of represent that so we can work with it. So in this case, we're going to use a capital B, and that's going to represent the dominant expression of the color, which is purple. So purple is the dominant color. So that allele, we will give a capital B to represent that purple is the dominant allele. The recessive allele is represented by a lowercase letter, and in this case, white is recessive. And so we're just going to use a little b. Now, it could be any letter that you use. In this case, we're just using big B and little b. Why it's not P, I don't know. I would be using P. There's purple, but that's okay. Anyways, so for this gene, you could have, each chromosome could have two, like you could have two purple genes, right? You always have two. You have two purple genes. You could have two genes for white, or you could have one purple and one white gene. Okay, so your combinations could look like this, right? And depending on the combination, you'll get a different color in the resulting plant. So if your plant has two of the dominant genes, remember the capitals represent dominant genes, and the dominant gene was purple flower, right? So if you have two dominant genes, you're going to get a purple flower. If you have one dominant gene and one recessive, so one purple, one white, well, we know the dominant is going to overpower the recessive, and you're not going to see that recessive. It's going to be hidden. So we're going to get a purple plant. So when we're looking at a purple plant, we can't tell if it's got two dominant alleles or one dominant and one recessive. I'm going to move my picture here because now we're going to look at there. We've got two recessive alleles, so two white alleles. All right. When we have two white alleles, we don't have the dominant one overpowering it. So that's the only time you're going to see the recessive trait of the white flower, when you have two recessive alleles. Okay, we're going to do quite a few more examples in this. So if you're not quite there with me, keep coming on the next few slides and we'll get there. Okay, so another dominant versus recessive trait that you can test out on yourself is, can you roll your tongue? I can. If you can, then you have at least one dominant allele. So we're going to represent tongue rolling. This makes more sense. If you can roll your tongue, a capital T. Okay. If you can't, that means you have to have two recessive alleles, little t's. Okay, so if you can't roll your tongue, you know that you have two recessive, one from your mom, one from your dad. If you can roll your tongue, you have to have one dominant allele at least, and the other one could be a dominant one. But the other option is you could have one dominant and one recessive. As long as you have a dominant allele in your combination, it's going to be expressed and the recessive will be hidden. All right, so we talked about Mendel in the last lesson. These are some of the traits that he studied and which ones were dominant versus recessive. And we looked at the purple and the white flowers. These are just a few out of interest sake. Seed color, yellow is dominant, green is actually recessive. Just out of interest sake, we don't need to memorize those or anything. Just kind of neat to see. And then in humans, here are a few of our traits. Oh, freckles is actually a dominant trait. No freckles is recessive. Brown eyes, blue eyes, there you go, dimples, no dimples. There's a few interesting ones there to check out. So it's also good to note that just because something says dominant doesn't mean that allele is very common in the population. Okay, you could have majority of the people just having recessive alleles, and then every once in a while that dominant allele comes out. Okay, and this is an example here. The most common form of dwarfism, this achondroplasia, is actually a dominant trait. So in your mind, you're thinking, oh, wow, if it's a dominant trait, it should be really popular. Well, it's not very common, right? And not very common at all. But it's because this dominant allele is not in a high concentration in the population. Okay, so majority of people have the recessive allele for this, so it doesn't come up in our genetic pool. Alrighty, some more terminology. Okay, so now we've talked about the different possible alleles and the way they group together. So we can have two dominant alleles and two recessive alleles together like that. See how when they're the same, so two big B's or two little B's. When we have identical alleles, we call this homozygous. Okay, or you might hear it referred to as purebred. Right, so they're not mixed up. The alleles are the same on both chromosomes for that person or that organism. You can have, as we've seen, one dominant and one recessive allele, and this situation is called heterozygous. So homo is the same, homozygous, the same. So you either are BB, two dominants, or two recessives. Okay, that's purebred. If you're heterozygous, that means you have one dominant and one recessive allele. All right, so that's some terminology you need to know, because in the problems you're going to get, they might just say you have a homozygous dominant female with a heterozygous male, and you need to be able to write down those alleles. So write down that terminology if it's not quite clear for you. Pause and do that. All right, genotype, when we're talking genotype, this is when we're looking at the actual genes, the allele combination. So everything what we've been talking up to right now. So for example, the genotype for a tongue roller could be big T, little t, which we call heterozygous, right? Heterozygous, or big T, which is homozygous dominant. And non-tongue rollers could have little t, which remember is homozygous recessive. So this is homozygous, ooh, bad spelling, recessive. Oh, and I went off the board. Okay, so when we're talking genotype, we are talking what do the alleles look like? That would be on the chromosomes. When we are talking phenotype, this is the outward expression of those alleles. So how is it going to appear to us when we look at the organism? So phenotype. So in this case, can you roll your tongue or can you not roll your tongue? That's the phenotype. Purple flowers or do you have white flowers? Do you have a widow's peak or no widow's peak? Hitchhiker's thumb, no hitchhiker's thumb. It's your physical appearance. So when you're asked for the phenotype, it's what the organism looks like, expressing that trait. And the genotype is when you're actually looking at what alleles that organism has. Again, two terms that you need to understand. So pause the video and write them down, write some examples down so you get that really clear in your head. All right, on to the next one. So we're looking at some more terminology here. Oh yeah, we're going to try using some of this terminology that we've just done. So we want to know what the phenotype of the following are. Oh, it's giving it away. I'm going to have to make up some new ones. So if you are given, when you're given two capital R's like this, let's go find them. Oh, there we go. There's the genes, the alleles that we're talking about. We're talking about seed form. And the allele for it is around, is the dominant. Okay, and in this case, we have two dominant alleles. When you have two dominant alleles, just one dominant allele actually is all you need, and your seeds will be round. Okay, so those are round seeds. If you have a capital Y and a little y. Let's go find there. There we have a allele for a yellow seed and we have an allele for a green seed. Remember, you only need one dominant one and it overpowers a recessive. So what is this going to look like? Because that's what we're trying to find. Phenotype, what does it look like? It's going to be a yellow seed because that capital Y is going to overpower it. Now, what if you have looks like two recessive alleles here, little g and little g? Down here, there's little g. Pod color is what we're talking about. It's recessive, but because there's no dominant one, we are going to actually have the recessive allele for the yellow pod color expressed. So that'll be a yellow pod color. Let's just try one more. Say you have L and little l, which is hard to, I'll do a little L like that. L and little l. This is our genotype I'm giving you, and I want to know the phenotype. Right, so I'm giving you the genotype, you tell me the phenotype. Pause the video and see if you can figure that out. All right, hopefully you paused this and you came up with this. So our the gene is for stem length. We're heterozygous, right? They're different. They're not the same. They're hetero. We have a tall allele and we have a short allele. Because we have one of each alleles, the dominant one is going to overpower. So our plant phenotype is tall. And that's all we need for phenotype. Okay, so what does the plant look like if we tell you the genotype? If we tell you what the genotype alleles are, you tell me what the phenotype, what it's going to look like. All right, let's do some more. So there's a little bit more terminology here, and then we'll try another couple of questions. So homozygous dominant. When you see homozygous, think homo means the same. Okay, that means your two alleles have to be the same, and dominant means they need to be the dominant alleles. So in case of tongue rolling, capital T, because you're talking about dominant and homozygous, it's always going to be two capitals in this case. All right, so it might be big B, homozygous dominant, always has to have two capitals. Homozygous recessive. Remember, recessive alleles we represent with lower cases, and homo again means same. So homozygous recessive means you're going to have two lowercase or two recessive alleles. So if we're talking about tongue rolling, two little t's. Heterozygous, hetero means different. Okay, as soon as you see heterozygous, you know you have to have one dominant and one recessive allele. All right, so in terms of tongue rolling, big T, little t. In terms of our B's, it would be big B, little b. And it's going to be expressed as this trait is, the phenotype will be whatever the dominant trait is. So whatever that big B is, or the big T. So tongue rolling in this case. Now, these individuals here, these heterozygous individuals, are called carriers because they have one of the recessive alleles in their cells. So they're able to pass on this recessive allele to their offspring. They might not ever, they'll never show it during their own lifetime, but they could pass it off to their offspring. And their offspring might show it, depending on what is the combination. So they're called a carrier of the recessive allele. Okay, and we talked about this last part about just because a trait is dominant, it does not mean the majority of the population are going to express that. Okay, we talked about that with the dwarfism example. All right, now Punnett squares. These are fun, actually, I think, and they're a fantastic tool to figure out what the offspring are going to look like, what the percentage and probability of what an offspring are going to look like when you know the genotypes of both parents. So it's a technique called using the Punnett square, and this British geneticist Reginald Punnett created the technique. And basically, you take the gametes from one parent and you arrange them along one side of the square, and then the gametes of the other parent along the other side. And then you do a little cross to see what kind of combinations are possible in the offspring, and then you can figure out the percentages. Now, we're only going to do this with a single trait in this course. If you've taken biology, you've probably done this with a couple traits, and it gets a bit more tricky, which is fun. But we're going to stick with what's called monohybrid crosses, or single traits. This video here too, if you're still a little confused after this lesson, make sure you check out that video. It's a great step-by-step explanation of how to use a Punnett square. Alrighty, so let's take a look at using a Punnett square. Let's go back to our purple and our white flower, which we know quite well now. So this purple flower, the genotype, that's this. Let's just use some of this terminology. Genotype, we can see, is heterozygous. heterozygous because it has a dominant and a recessive allele. Now, because it has one of the dominant alleles, which is the purple color, is dominant, it is expressed as a purple flower. So its phenotype is purple. Okay, so what we're going to do when we use our Punnett square is, oh, I started this backwards, but that's okay. I'll stop here at this point. We're going to take its possible gametes. So remember, when you form gametes in meiosis, you take only half of the genetic material to the egg cells or to the sperm cells. So that means when you create an egg or a sperm cell, it's only going to have one of these alleles. So it's going to have either the purple dominant allele or the white recessive allele in that case of that flower. And we put each of these possible gametes alleles along the cross of the top of the Punnett square. Okay, I'm going to leave that one right for now because I started on the wrong side here. I don't know why I started on the right-hand side. We always read left. Anyway. Okay, so we're going to now take a look at this white flower, right? So white, remember, is the phenotype. And it's got two recessive alleles. That is its genotype. So I'm using that terminology. And they're both the same. There's two recessive, two same, which means it's homozygous. And it's homozygous recessive. Okay, so when this flower, this white flower, forms gametes, it can only form gametes that have small p's, or the recessive allele. It doesn't have any of the dominant alleles in it, or the capital P's. So all of its egg or sperm cells, or gametes, are going to be little p's. So they go along the top of the Punnett square box. Now, we're going to take this white flower here, and we're going to cross it, mate it, if you mate flowers. Anyway, we're crossing it with the purple flower here. This purple flower, you can see it's got two of the capital P's, or two of the dominant alleles for flower color. And again, so it can only, this flower, when it makes seeds, or gametes, it can only have capital P's in its gametes. So you're going to have a capital P and a capital P. So when these two flowers are crossed, we can see what the combinations are going to be. So if we used this gamete and this gamete, we're going to get a capital P and a little p. And this gamete and this gamete, again, capital P, little p. You start to see a pattern here. They're all of these offspring are going to be exactly the same. They're all going to be heterozygous, right? 100% heterozygous. There he goes. 100%. This is our genotype. 100% big P, little p. Heterozygous. And if we're talking about phenotype, the phenotype is purple. They're all going to be purple. All right, so that's how Punnett square lets us figure out what are the offspring going to look like? Well, that was an easy one because they're all going to look the same. So let's take one of these purple flowers, one of these offsprings. Here we go. And we're going to do another cross. And we're going to try and figure out what the next generation looks like, which is referred to as the F2 generation. So we're going to take those same purple flowers. So we have a dominant allele and we have a recessive allele. That purple flower can only give a dominant allele or a recessive allele when it's forming gametes. And then we've already talked about this purple flower up top here. This was homozygous, recessive. Nope, sorry, heterozygous. So it has one dominant allele and one recessive allele. And we're going to do a cross here and see what we get. And this time we're going to get a little something different. So here we have the dominant allele combining with a dominant allele. So we've got two dominant alleles here, which are going to give us a purple flower. Here we have a recessive allele and the dominant allele, but we still have a dominant allele, so it's still going to be purple. We have the dominant allele coming down, and we have the recessive allele. So it's still going to be purple. And then we have a recessive and a recessive. So here we have homozygous recessive. So in this case, there's no dominant allele hiding the recessive trait. So this is our plant that's going to be white. So if we look at the population of all the possible offspring, 75% of them will be purple for when we're talking phenotype. 25% will be white when we're talking phenotype. Let's talk genotype though. So genotype's slightly different, right? We want to look at the actual alleles. So if we want homozygous dominant, what percentage of the offspring are homozygous dominant? Well, your homozygous dominant, that's the only one there that's homozygous and dominant. So that's 25% of the offspring. Okay, what percentage are homozygous recessive? Homozygous recessive. 2 p's, 2 little p's, right? There's 25%. So what's the genotype of the other 50%? Heterozygous. Big P, little p. Heterozygous. 50% are heterozygous. All right, so there's an example worked through on how we use the Punnett square to figure out the percentage of what the offspring are going to look like, right? This just gives us a probability. It doesn't always give us the exact amount. It's just kind of like the probability of if you were to reproduce, what are the chances of having this certain combination? All right, so just to recap, there's the procedure on how to do a Punnett square. First, you're going to write down the letters that you would use to indicate what's going to be dominant and what's recessive. So if it's not given to you, come up with what you're going to use as your dominant and recessive alleles. So make sure the dominant is the capital and your recessive is the lowercase letter. Then you write down the genotypes for what the parents look like, for what the parents have. Okay, so if they call this a homozygous dominant, that's what it would look like. And this would be your homozygous recessive parent. All right, and then you want to show what the different gametes from each parent will produce. So what gametes are each parent going to produce? And these parents can only form gametes that have capital T's, and this parent here can only form gametes with lowercase t's. If you happen to have a heterozygous parent, your gametes that would form would be 1 capital, 1 little t. Those are your only options. Okay. And then you're going to use a Punnett square to show the results. And that's on the next slide. And then state the phenotype and genotype ratios. All right, so we took our parents, our capital T parents, put it along the top. So our capital T, or our homozygous dominant parents, alleles. So these are the 2 possible gametes that it can form. And then there is our homozygous recessive parent and its possible gametes that it forms. Then we use our Punnett square to figure out what combinations we get, right? Remember, you always have to get an allele from a parent, a chromosome from a parent that contains that allele and a chromosome from the other parent that contains the other allele. So this gives us our mixture here. And then there's our capital T, here's our little t, here's our capital T, here's our little t, capital T, little t. So this lets us mix all these alleles up and figure out what our combination is. So when we look at all the offspring have the exact same genotype. When we're talking genotype, the alleles, they're all heterozygous. When we're talking phenotype, they're all going to be tall, 100% of them tall. Okay, so there's a genetic worksheet that we're going to be working on, and there's actually a video of all the solutions for that worksheet. So it's really handy if you're having any struggles. I'm going to, oh yeah, there's one more little spot here, but I think we've done quite a few. I don't want to overdo it. This is an example of a Punnett square with the probability of beans using either green or yellow, with one parent has 2 recessive traits. Okay, so if you're feeling comfortable with Punnett squares, you can go ahead and start working on some of the practice questions. If you want to do a little bit more, then continue watching. If this is new to you and you're like still wondering what's going on, let's use some more of the terminology to keep it in your head. Okay. So in this case, you can see, let's just look at the parents here. There's 2 little y's, right? So how would you describe the genotype of that parent? You can pause the video and see if you can describe the genotype of each of these parents. Okay, so genotype is just going to be, it's that little y. So this is homozygous recessive. And this genotype is heterozygous. Hetero. Okay. And you can see what color they look. So green pods are the recessive trait. So this has a phenotype of a green pod, and this over here has the phenotype of the yellow pod. It's expressing the dominant gene. We do the cross. So I love this one because the colors, you can see it a little bit easier where the alleles are coming from. Aha, this gives us a little different variety in this case. So all of the offspring, so there is look at that, half of the offspring are going to be homozygous recessive or green, have the green phenotype for the green pods. So 50% of them are going to be like that. And the other 50%, because they're exactly the same, these two, the other 50% are heterozygous. And because they have one dominant allele, they are going to express that dominant allele, so they will be yellow pods. All right, so you can review this eye color question and see if you can get it. Read the top part, cover up the answer, see if you can figure out the Punnett square yourself, and then check with your answer down below. So I'm going to pause this video and we're going to start a new video for sex-linked traits, because this is a lot of information for some people who've never done any of this genetics before, and I want you to work with Punnett squares before we start looking at the X and Y chromosomes, because they carry traits all by themselves and have some other unique things that they're involved in. All right.